Question 1. Which property of carbon most contributes to the diversity of organic molecules? A. It readily forms ionic bonds with most elements. B. It has 4 valence electrons and can form single, double, and triple covalent bonds in chains, branches, and rings. C. It can donate protons to act as an acid. D. It rarely forms covalent bonds with itself. Correct answer, B. Carbon's tetravalence allows it to form up to 4 covalent bonds and build chains, branched skeletons, and rings with single and multiple bonds, creating extraordinary structural diversity in biomolecules. Question 2. Which interaction is primarily responsible for water's cohesion and surface tension? A. Hydrogen bonds between water molecules. B. Covalent bonds within a water molecule. C. Ionic attractions between sodium and chloride ions. D. Only van der Waals interactions. Correct answer, A. Hydrogen bonds form between the partially positive hydrogens and partially negative oxygens of neighboring water molecules, producing strong cohesion, surface tension, and capillary action in biological systems. Question 3. Which element is part of the backbone linkage that connects nucleotides in DNA and RNA? A. Carbon. B. Nitrogen. C. Phosphorus. D. Sulfur. Correct answer, C. Phosphorus is in the phosphate group that forms phosphodiester bonds between the 3' hydroxyl of one sugar and the 5' phosphate of the next, creating the sugar-phosphate backbone of nucleic acids. Question 4. Which statement best explains nitrogen's essential role in proteins? A. Nitrogen forms the amino group and participates in peptide bonds linking amino acids. B. Nitrogen stabilizes carbohydrate rings with ionic bonds. C. Nitrogen creates disulfide bridges between cysteines. D. Nitrogen is the primary cellular energy source. Correct answer, A. Nitrogen is part of amino groups in amino acids and is incorporated into peptide bonds during dehydration synthesis, making it indispensable for protein structure, unlike carbohydrates, which contain only C, H, and O. Question 5. Which interaction stabilizes tertiary structure by forming covalent links between cysteine residues? A. Ionic bonds between charged R groups. B. Hydrogen bonds along the backbone. C. Disulfide bridges formed by oxidation of sulfhydryl groups. D. Hydrophobic interactions among nonpolar side chains. Correct answer, C. Sulfur-containing cysteine residues can oxidize to form covalent disulfide bridges, locking parts of the polypeptide together and strengthening tertiary structure, especially in extracellular proteins. Question 6. What type of reaction builds biological polymers from monomers? A. Hydrolysis. B. Oxidation. C. Dehydration, also called condensation. D. Hydrogenation. Correct answer, C. Dehydration reactions remove a molecule of water to form new covalent bonds, glycosidic linkages, peptide bonds, and phosphodiester bonds, assembling monomers into polymers. Question 7. Which part of a phospholipid interacts most strongly with water? A. Long fatty acid tails. B. Glycerol backbone. C. Phosphate-containing head group. D. Cholesterol ring system. Correct answer, C. The polar, negatively charged phosphate head is hydrophilic and hydrogen bonds with water, while the nonpolar hydrocarbon tails are hydrophobic and avoid water. Question 8. Why can humans digest starch but not cellulose? A. Human enzymes hydrolyze alpha-1,4 glycosidic linkages in starch, but cannot hydrolyze beta-1,4 linkages in cellulose. B. Cellulose dissolves readily in water. C. Cellulose contains nitrogen atoms. D. Starch is unsaturated while cellulose is saturated. Correct answer, A. Amylase recognizes the alpha orientation in starch, but humans lack enzymes for the beta-1,4 linkages of cellulose, so cellulose is dietary fiber unless microbes provide the enzymes. Question 9. Which change increases membrane fluidity at low temperature? A. Increase saturated fatty acids. B. Lengthen fatty acid chains. C. Add trans double bonds. D. Increase cis unsaturated fatty acids. Correct answer, D. Cis double bonds introduce kinks that prevent tight packing of phospholipid tails, maintaining membrane fluidity when it's cold. Question 10. Which components are found in a nucleotide? A. Amino group, carboxyl group, and R group. B. Glycerol, phosphate, and 2 fatty acids. C. Phosphate, 5-carbon sugar, and a nitrogenous base. D. Steroid nucleus and hydroxyl group. Correct answer, C. Each nucleotide consists of a pentose sugar, a phosphate group, and a nitrogenous base. Nucleotides polymerize via phosphodiester bonds to form nucleic acids. Question 11. Which functional group can donate a proton and is acidic near physiological pH? A. Carboxyl. B. Amino. C. Sulfhydryl. D. Carbonyl. Correct answer, A. Carboxyl groups can release H+ to form carboxylate, acting as weak acids. Amino groups generally act as bases by accepting H+. Question 12. Which element's high electronegativity in water creates polar covalent bonds with hydrogen, enabling hydrogen bonding? A. Carbon. B. Nitrogen. C. Oxygen. D. Phosphorus. Correct answer, C. Oxygen pulls shared electrons toward itself, giving water partial charges that allow extensive hydrogen bonding and unique properties like high specific heat and cohesion. Question 13. Which level of protein structure is defined by the linear sequence of amino acids linked by peptide bonds? A. Secondary. B. Tertiary. C. Quaternary. D. Primary. Correct answer, D. Primary structure is the amino acid sequence connected by peptide bonds. It dictates higher level folding and ultimately function. Question 14. Which statement correctly distinguishes ribose from deoxyribose? A. Ribose lacks an oxygen at the 2' carbon. B. Ribose has a hydroxyl at the 2' carbon that deoxyribose lacks. C. Ribose has 1 fewer carbon than deoxyribose. D. Ribose lacks a 5' carbon. Correct answer, B. Ribose has an OH at the 2' position, whereas deoxyribose has H there. The extra hydroxyl makes RNA less chemically stable and more reactive than DNA. Question 15. If a cell is phosphorus limited, which biomolecules would most directly decrease in abundance? A. DNA and RNA. B. Polypeptides and disulfide bonds. C. Glycogen and starch. D. Cellulose and chitin. Correct answer. A. Phosphorus is required for nucleotides, ATP, and phospholipids. Phosphorus limitation reduces nucleic acids and energy currency first, not carbohydrate polymers or peptide bonds. Question 16. Which macromolecule is not a true polymer built from repeating monomer subunits? A. DNA. B. Proteins. C. Lipids. D. Starch. Correct answer. C. Lipids such as triglycerides and phospholipids are assembled, but not from repeating monomer units in long chains like nucleic acids, proteins, and polysaccharides. Question 17. Which side chain property most promotes formation of a protein's hydrophobic core? A. Charged R groups. B. Polar uncharged R groups. C. Nonpolar hydrocarbon R groups. D. Aromatic rings only. Correct answer C. Nonpolar side chains aggregate away from water via hydrophobic interactions and van der Waals forces, driving the collapse of the protein interior. Question 18. Which property allows large bodies of water to resist rapid temperature change? A. Low specific heat. B. High specific heat due to hydrogen bonding. C. Low heat of vaporization. D. High compressibility. Correct answer B. Heat energy is absorbed to break hydrogen bonds before water's temperature rises, buffering ecosystems and stabilizing organismal temperatures. Question 19. How does a biological buffer, such as the bicarbonate system, resist pH change? A. It neutralizes all incoming acids. B. It holds hydrogen ion concentration constant. C. It reversibly accepts H+ when acid is added and donates H+ when base is added. D. It converts strong acids to bases. Correct answer C. Conjugate acid-base pairs buffer solutions by shifting equilibrium to bind or release H+, minimizing swings in pH around a set point. Question 20. Two molecules have the same molecular formula but differ in spatial arrangement around a double bond. These molecules are best described as A. Structural isomers. B. Enantiomers. C. Cis-trans or geometric isomers. D. Isotopes. Correct answer C. Double bonds restrict rotation, so groups can be on the same side, cis, or opposite sides, trans. Differences that change molecular shape and biological activity. Question 21. A peptide bond forms between which functional groups during protein synthesis? A. Two sulfhydryl groups. B. The amino group of one amino acid and the carboxyl group of another. C. Two phosphate groups. D. Two hydroxyl groups of adjacent sugars. Correct answer B. During dehydration synthesis, the carboxyl of one amino acid reacts with the amino of the next, releasing water and forming a covalent peptide bond. Question 22. Which DNA sample would have the highest melting temperature? A. One with higher GC content. B. One with higher AT content. C. One with longer Okazaki fragments. D. One with more uracil bases. Correct answer A. GC pairs form 3 hydrogen bonds versus 2 in AT pairs, so GC-rich DNA requires more thermal energy to separate strands. Question 23. Which change would most directly increase a membrane's permeability to water? A. Replace phospholipids with triglycerides. B. Greatly increase cholesterol content. C. Insert aquaporin channel proteins. D. Shorten fatty acid tails. Correct answer. C. Aquaporins are specialized channel proteins that allow rapid regulated water transport across cell membranes. Question 24. Which statement best describes cholesterol's effect on membrane fluidity? A. It always increases fluidity. B. It always decreases fluidity. C. It has no effect on fluidity. D. It reduces fluidity at moderate temperatures but hinders solidification at low temperatures. Correct answer, D. Cholesterol acts as a fluidity buffer, restraining phospholipid movement when warm and preventing tight packing when cold, keeping membranes functional over temperature changes. Question 25. Which property of oxygen makes it effective as a terminal electron acceptor in biological systems? A. Low electronegativity makes it easy to reduce. B. High electronegativity allows it to pull electrons down the electron transport chain, releasing free energy. C. It forms ionic bonds with protons. D. It donates electrons to NAD+. Correct answer, B. Oxygen's strong affinity for electrons makes redox reactions highly favorable, enabling the electron transport chain to harness energy for ATP synthesis.